The rate of movement of substances across the cell membrane is determined by the concentration gradient of the substance being exchanged and the size and shape of the cell. Rate refers to how rapidly a substance can move across the membrane, whether it's faster or slower. Diffusion relies on the relative concentration gradient of a substance on either side of a membrane. If the concentration gradient is high, then substances will diffuse rapidly across the membrane. As the concentration becomes more similar on either side of the membrane, the rate of diffusion will become slower. Cells can actually do various things to increase the concentration gradient across the membrane in order to maintain a rapid diffusion rate. For example, plant cells use a process called cytoplasmic streaming. This is where organelles and the cytosol move in a circular motion inside the membrane. This means that materials that are diffusing into the cell are rapidly removed. This helps to maintain a steep concentration gradient, particularly in comparison to if the cytoplasm was stationary. Multicellular animals can maintain steep concentration gradients by removing some of the substance that is being diffused across the membrane. For example, when you breathe in oxygen, it diffuses into the cells lining your lungs. That oxygen is then rapidly distributed throughout your body by red blood cells. Another method is to convert the substance being diffused into another molecule, therefore lowering its concentration. For example, glucose molecules diffuse into liver cells and are used up or are converted to glycogen. This maintains a steep concentration gradient for glucose to diffuse into your liver cells. Another important concept that affects the rate of exchange of materials across membranes is the surface area to volume ratio. And let's start by taking a look at this concept in a cube rather than a cell. If you unfolded a cube, you would end up seeing the surface area, which would be the entire outside surface of the cube. And if this cube was 1 centimeter on each side, one side would be 1 centimeter squared in area. Now, because there are 6 sides, the entire surface area of the cube is going to be 6 centimeters cubed. Now, on the other hand, the area is the entire volume of that cube, which in this case would be length by width by height, which would be 1 centimeter cubed. Now, if you were to express this as a ratio of surface area to volume, this would be 6 to 1. And that is what's known as the surface area to volume ratio. This can also be represented as a number if you divide the surface area by the volume. In this case, it would be 6. So cells don't really resemble cubes, so let's try spheres, which they look more like. So if you were to calculate the surface area and volume of 3 different size spheres, one being 3 centimeters in diameter, another being 2 centimeters in diameter, and the last one being 1 centimeter in diameter, what you would find is that the 3 centimeter diameter cube would have a surface area of 28.28, and then respectively 12.57 and 3.14 centimeters squared. Their volumes would be 14.14 centimeters cubed, 4.19 centimeters cubed, and 0.52 centimeters cubed. The ratio between the surface area and volume for each of these spheres would then be 2 to 1, 3 to 1, and 6 to 1. And what is the relationship you notice between the size of these spheres and their surface area to volume ratio? Well, if you notice, as the cells decrease in size, their surface area to volume ratio increases. And this is very important. The uptake of materials from the external environment into a cell occurs via its cell membrane. These materials are then used to fuel the chemical reactions that occur throughout the volume of the cytoplasm. For a cell to be able to supply the cytoplasm and its metabolic requirements and remove wastes, you need a large surface area in relation to the volume of the cell, or a large surface area to volume ratio. This is the reason why cells tend to be very small. It enables them to have a large surface area to small volume, and this enables substances to diffuse into the cell at a more efficient rate than if the cell was larger.